everyone. Today we will discuss about photosynthesis in higher plants. This is chapter number 11 from NCERT Biology class 11th. So photosynthesis is a basic process which is required to sustain the life on earth, especially the green plants which has chlorophyll pigment. It produces its own food through photosynthesis. That's why it is called as autotrophs. So photosynthesis is a physiochemical process. Photosynthesis involves both physiological and chemical process. That's why it's called as physiochemical process. So as we already know, for photosynthesis, we need carbon dioxide, water in the presence of sunlight, and chlorophyll. So, so the physiological process in photosynthesis is water uptake and carbon dioxide uptake. So carbon dioxide is taken inside by the plant to stomatal pore and water through the roots. These are all physiological process. The chemical process is light reaction, CO2 fixation, breaking of water to hydrogen and electrons. So all other and fixation of CO2 to starch, conversion of CO2 to carbohydrates. These are all chemical process. That's why it's a physiochemical process. So let's understand about what is really photosynthesis. For definition, if you go for photosynthesis is a process by which CO2, carbon dioxide, and water is converted to carbohydrates in the presence of sunlight and chlorophyll. So it requires sunlight and chlorophyll. That's why it's called as photosynthesis. Photo is nothing but sun. Synthesizing light energy. Synthesizing photo is called photosynthesis. So the light energy is converted into chemical energy. The light energy is synthesized into chemical energy. That's why it's called as photosynthesis. So CO2 and water is converted to carbohydrates, that is glucose, in the presence of sunlight and chlorophyll. So if you go by chemical formula, the chemical equation, so 6 molecules of CO2 reacts with 12 molecules of water in the presence of sunlight and chlorophyll to give 1 molecule of glucose, that's C6H12O6, plus 6 molecules of water and 6 molecules of oxygen is evolved. So important steps in photosynthesis. The first step is the chlorophyll, that's a green pigment present in the leaf. It traps the sunlight. So it needs an energy. So it comes from the sunlight. Sunlight as a photons. That's the energy source. So for everything which we want to happening in Earth, in whole planet, sunlight is the primary energy. So for photosynthesis, sunlight is the primary energy. So chlorophyll in the leaf, which is a green pigment, which traps the sunlight, which absorbs sunlight, that's the light energy. This light energy later converted into chemical energy, that is through chemical process, the carbon dioxide fixation. So and also the water is broken down to hydrogen, oxygen, and 4 electrons. So electrons is very much required to maintain photosynthesis process. So later the CO2 is reduced to carbohydrate. I mean CO2 is fixed to carbohydrates, that's glucose. Why it is reduced? There is an addition of hydrogen here. So addition of hydrogen is called as reduction. So CO2 is converted into reduced into carbohydrates. That's why it's called reduced. So next going to section 13.1. So what do we know in the photosynthesis previously? That's chapter 5th in class 10th. So there are some experiments to understand the photosynthesis process. The first experiment was done with the starch and iodine reaction. That's called a starch iodine test. So this experiment was initially done to verify whether chlorophyll or green part is essential for photosynthesis or not. But we already know that the green part has the chlorophyll, so which absorbs the light energy, and that's where the photosynthesis happens. But earlier in 17th century, they don't know anything. So they wanted to check whether chlorophyll, the greener part in the leaf, is really required for photosynthesis. So what they done, they've taken a plant with a variegated leaf. So variegated leaf means a leaf which is having different colors. So green, the whatever you see, the shade, the colored with the some part of the leaf is greener, some part of the leaf is white, some part of the leaf is yellow. So that's why it's called variegated leaf, like a mosaic leaf. So the greener part has a chlorophyll in that. So other part doesn't have chlorophyll in that. So only the greener part has chlorophyll in that. So what they have done, they have taken the variegated plant and they kept in the dark to remove all the starch which is produced through photosynthesis previously. To remove it from the plant, they kept the plant in the dark condition for 3 days. So there is a zero starch now. So then later they have shifted it to sunlight. In the sunlight, after keeping in sunlight for several hours, they took the leaf from that plant and they put it in boiling water, and they also put it in alcohol. So why they have done this? Just to dissolve chlorophyll. They want to do the staining with iodine. So chlorophyll is already a greener pigment. So you can't see the iodine pigmentation properly. That's why they wanted to remove all the chlorophyll pigment. So the experiment is already over. That's why they want to remove all the chlorophyll pigment. So when this leaf is treated with iodine, so iodine in normal is orange to brown in color. But when this iodine is treated with the plant, the leaf is treated with iodine. So the iodine reacts with the starch, and only the areas where the green pa- part was there in the leaf were turned into deep blue. How it turned into deep blue? So iodine reacts with starch. Starch has amylose sugar and amylopectin sugar. So amylose has a helical structure. So in this helical structure, the iodine gets binded and it's give blue color. So wherever the green part is there, green region is there, there it has the blue staining color, deep blue staining color. That means wherever the green region is there, there only the starch has been produced. So starch is produced, that's why it's reacted with iodine, it's giving deep blue color. So iodine is at normal as a orange brown color. When it reacts with starch, it produces deep blue color. So what it indicates, only the region where the green color, the chlorophyll or green pigment is there, that part only is producing starch. So starch is produced through photosynthesis. So that's why it indicates chlorophyll is essential or green part is essential for photosynthesis. This is proven by starch iodine test. So another test is KOH experiment, potassium hydroxide experiment. This experiment is done to check whether CO2 is essential for photosynthesis or not. So we don't know, earlier they don't know anything about photosynthesis. They wanted to check what is really required for photosynthesis, whether CO2 is required, oxygen is required, whatever. And so this test was done to whether CO2 is required for photosynthesis or not. So they have taken two plants, plant 1 and plant 2 with leaves, and the same thing, they are kept in dark room to remove all the starch. And again, after keeping in dark room for 3 days, Why they want to the keeping in dark room? So they want to make sure that there is no previous starch left. So they are making an experiment. So that's why they are making it zero. So they are making zero starch, and later one plant is kept in a closed jar, other plant is also kept in a closed jar, but plant one inside the closed jar they have kept KOH substance, KOH chemical. So what KOH chemical does? KOH is a basic substance. It absorbs or it reacts with acidic, that's the CO2, because inside the air is there. So normally air has a CO2 and oxygen and everything. The CO2 is there. This KOH absorbs all the CO2 and it make potassium carbonate and water. So it's a basic, and carbon dioxide is acidic. So basic acidic reaction. So it absorbs all the carbon dioxide and the carbon dioxide is not available to the plants. Here there is no KOH, so carbon dioxide available to the plant. So it's a tightly sealed. There's a air packed sealed. There is no air going. There is no air coming from outside. So whatever the air is inside, only that much is available for the plant. But here the KOH is kept. All the CO2 is taken down by the KOH. So again, when they are kept in the sunlight, and again when they are treated with iodine test, only the plant 2 showed starch. That's a deep blue color. Why? Because there is a CO2 is available here. But plant 1, there was no starch and there is no blue color. Why? The CO2 is absorbed by KOH. That's why plant can't use CO2 to produce carbohydrates or the starch. That's why it proves that the carbon dioxide is required for starch formation. The starch formation happens through photosynthesis. Thank you. Hello everyone. So today let's start with section 11.3 from chapter number 11, NCERT Biology class 11th, photosynthesis in higher plants. So section 11.3 speaks about the location of the photosynthesis. Where does photosynthesis takes place? So photosynthesis takes place in any greener parts of the plant, including green leaves, the other green parts like green stems, young fruits, sepals, green floral parts. So the neat important point for the neat is any green part containing chloroplast can perform photosynthesis. Not only green leaves, but any parts which is greener can perform photosynthesis. Sometimes the stems looks greener, right? And the young fruits looks greener, sepals looks greener. So all the green parts can perform the photosynthesis. This is very important. Especially the chloroplast is present in the mesophyll cells. So mesophyll cells contain the maximum amount of chloroplast. So this is very important cells. The mesophyll cells, which is next to the epidermal cells, contains the chloroplast. That's where the real photosynthesis happens. So the chloroplasts are aligned. The alignment of chloroplast is very important. The chloroplasts are aligned in the mesophyll cell along the cell's walls. Why? This helps them receive maximum light. So the alignment of the chloroplast with respect to the cells differs according to the light intensity. When there is a low intensity, the low light or the low intensity, the cells needs a lot of light to be trapped. That's why the chloroplast align flat surface. The flat surface of the chloroplast aligns perpendicular to the light, so it can absorb the maximum amount of light. But during the high light intensity, the flat surface of the chloroplast aligned parallel to cell wall. You can see here during the high light intensity, this is the flat surface is aligned parallel to cell wall. Here at the low light, the chloroplasts are aligned perpendicular. See, if you see the perpendicular angle, this is 90 degree, perpendicular to the light direction. And in the moderate light, it appears like freely, it's floating everywhere. So they do call different terminology for different alignment of the chloroplast. During the high light intensity, when the chloroplast aligned parallel to the cell walls, they called as periscope. When it is aligned perpendicular to the light, it's called epistrope. When it is aligned free floating everywhere, mixed and is floating everywhere randomly, it is called as apostrophe. So this is very important for the NEET. Why? To optimize, see, light, if the intensity of the light becomes more, it also causes damage to the chloroplast. So that's why the chloroplast need to be taken care itself, so that the damage should not be occurred by the high light intensity. To avoid the high light intensity, to avoid the high damage occurred by the light, to optimize the light absorption, the movement of the chloroplast with respect to the mesophyll cells will be decided. सो एक टॉपिक समझ में आ गया. तो लो लाइट इंटेंसिटी में कैसा क्लोरोप्लास्ट अलाइनमेंट होता है, हाई लाइट इंटेंसिटी में कैसा क्लोरोप्लास्ट अलाइन होता है, समझ में आ गया. फिर स्ट्रक्चर ऑफ क्लोरोप्लास्ट. तो क्लोरोप्लास्ट में क्या क्या रहता है? क्लोरोप्लास्ट में तीन तीन इंपॉर्टेंट चीजें रहता है. देयर आर थ्री थिंग्स व्हिच आर वेरी इंपॉर्टेंट. वन इज ग्राना. तो ग्राना, ये ग्राना बोलते हैं. तो ग्राना क्या मेक इज मेड ऑफ अ फुल ऑफ thylakoids. Each part is a thylakoids. Thylakoids is a membranous structure. You need to very important. Grana is a membranous structure. Chloroplast itself is a double membrane structure. देखो ये क्लोरोप्लास्ट है, तो एक आउटर मेंबर रहेगा, एक इनर मेंब्रेन रहेगा, और इसके अंदर ग्राना रहेगा. ग्राना रहेगा, स्ट्रोमा रहेगा, ये स्ट्रोमा लैमेला रहेगा. तो ग्राना का है, थैलेकॉइड मेंब्रेन का लेयर बाय लेयर एक के ऊपर एक के ऊपर जो स्टैकिंग हुआ है उसको ग्राना बोलते हैं. और ये ग्राना को दूसरा ग्राना को, देखो ये ग्राना हो गया, ये ग्राना, दूसरा ग्राना को जो कनेक्ट कर रहा है उसको बोलते हैं स्ट्रोमा लैमेले. इंटरकनेक्टिंग मेम्ब्रेन्स, दिस इज आल्सो मेम्ब्रेनियस स्ट्रक्चर. दिस इज आल्सो मेम्ब्रेनियस स्ट्रक्चर. एंड स्ट्रोमा, ये छोड़ के बीच में जो भी कुछ भी है, this liquid matrix is called as stroma. So this is very important. The chloroplast is a double membranous structure. आउटर मेम्ब्रेन, इनर मेम्ब्रेन. क्लोरोप्लास्ट के अंदर क्या रहता है? ग्राना रहता है. ग्राने के अंदर क्या रहता है? थैलेकॉइड स्टैक्ड रहता है एक के ऊपर एक के ऊपर. दो ग्राना थैलेकॉइड्स को अटैच करने वाले को क्या बोलते हैं? स्ट्रोमा लैमेला बोलते हैं. और जो फ्लूइड है उसको स्ट्रोमा बोलते हैं, द मैट्रिक्स. सो हर पार्ट का एक एक डिवीजन ऑफ लेबर रहता है. सो देयर इज अ डिफरेंट the capabilities and different work each part has. So सपोज थाइलाकॉइड मेम्ब्रेन और ग्राना क्या आता है? ये लाइट रिएक्शंस में इन्वॉल्व रहता है. थाइलाकॉइड के अंदर लाइट रिएक्शन, दिस इज अ फोटोकेमिकल रिएक्शन. दिस इज वेरी इंपॉर्टेंट. So light reaction is a photochemical reaction. Why? It's by using the light, we are breaking water and we are trapping the light energy. That's why इधर लाइट का यूज रहता है, इसलिए लाइट रिएक्शन को फोटोकेमिकल रिएक्शन बोलते हैं. It traps light energy and also synthesis of ADP, ATP and NADPH. स्ट्रोमा में क्या होता है? डार्क रिएक्शन होता है. इसका इस आल्सो कॉल्ड एज केल्विन साइकिल और कार्बन रिएक्शंस. क्यों? इधर जो लाइट रिएक्शंस का आउटपुट रहता है एटीपी एनएडीपीएच, उसके आउटपुट के ऊपर ये रिएक्शंस होता है. मतलब CO2 फिक्सेशन होता है, शुगर सिंथेसिस होता है और स्टार्च में होता है. इसको डार्क रिएक्शंस बोलते हैं. ऐसा नहीं है कि स्ट्रोमा में डार्क रिएक्शन लाइट जब लाइट है तब नहीं होगा, ऐसा नहीं होता है. Only it requires the outputs of light reactions to happen dark reaction. तो समझ में आ गया? थैलेकॉइड मेम्ब्रेन ग्राना में लाइट रिएक्शंस होता है, स्ट्रोमा में डार्क रिएक्शन होता है. डार्क रिएक्शन इज आल्सो कॉल्ड एज कार्बन रिएक्शंस और केल्विन साइकिल. लाइट रिएक्शंस इज आल्सो कॉल्ड एज फोटोकेमिकल रिएक्शंस. तो लाइट रिएक्शन में एटीपी और एनएडीपीएच का सब प्रोडक्शन होता है. और डार्क रिएक्शन में जो भी इसको यूज करके जो CO2 है उसको फिक्सेशन करके शुगर और स्टार्च प्रोड्यूस होता है. चलो लास्ट वीडियो में हम पढ़ लिया. सो क्लोरोप्लास्ट के अंदर क्या क्या रहता है. नाउ we are just need to discuss what is the function of part each each part of the chloroplast. So chloroplast के अंदर क्या करता है? each structure, ग्राना है, स्ट्रोमा लैमेला है, स्ट्रोमा मैट्रिक्स है. सो क्या क्या होता है? सो ग्राना थैलेकॉइड मेम्ब्रेन, जिससे stacked thylakoid membranes is called as grana. The grana is the place of light reaction. That's also called as photochemical reaction. क्यों photochemical reaction बोलते हैं? Because light reactions is happen only in the presence of light, which breaks water into hydrogen and electrons and oxygen. Oxygen is evolved. That's why it involves the light. That's why it's called as a photochemical reaction. लाइट एनर्जी को ट्रैप करके इलेक्ट्रॉन ट्रांसपोर्ट चेन को एक्टिवेट करके एटीपी और एनएडीपीएच का प्रोडक्शन होता है. कहाँ? लाइट रिएक्शन में. कहाँ होता है लाइट रिएक्शन? ग्राना में. इधर होता है लाइट रिएक्शन. लाइट रिएक्शन इधर होता है. डार्क रिएक्शन कहाँ होता है? ये ग्रीन कलर जो जितना भी मैट्रिक्स है, फ्लूइड है, ये सब इधर सब डार्क रिएक्शन होता है स्ट्रोमा में. डार्क रिएक्शन में क्या होता है? व्हाट हैपेंस इन द डार्क रिएक्शन? So whatever the ATP and NADPH which is produced in the light reaction, they got involved, they are utilized in the dark reaction. So what it happens, they fix the CO2, atmosphere CO2 लेके, वातावरण से एनवायरमेंट से CO2 लेके उसको ग्लूकोज या स्टार्च के, ग्लूकोज के बाद स्टार्च होता है, ग्लूकोज का इसमें फिक्स करता है. और फिक्स करने के लिए उसको एटीपी और एनएडीपीएच चाहिए. ये होता है डार्क रिएक्शन. डार्क रिएक्शन में क्या आता है? इट टेक्स द प्लांट टेक्स कार्बन डाइऑक्साइड फ्रॉम द एनवायरमेंट and it fix carbon dioxide to carbohydrates, that is glucose. Okay, that's a reduction reaction. I just discussed about all these stuffs, the equation of the photosynthesis in the first video, how CO2 is reduced to carbohydrates, that's the glucose. So the need के लिए क्या क्या important है? So light reactions is a photochemical reaction. Dark reaction is also called as carbon reactions or Kelvin cycle. So dark reactions do not occur in darkness. It doesn't mean that dark reactions only occurs in darkness. Dark reactions, they are dependent on the ATP and NADPH produced in the light reactions. So dark reactions is entirely dependent of the output of the light reaction. That's why it's called as a dark reaction. It doesn't mean that dark reactions happens only in the absence of light. No, it can happen anytime, but to happen the dark reaction, it should have the outputs of light reaction, that is ATP and NADPH. So it's a quick comparison between light reaction and the dark reaction. So in the light reaction happens in the thylakoid membrane, that's the grana. Dark reaction happens in the stroma. Light needed, directly light needed. Dark reaction में क्या होता है? Indirectly, because it doesn't need light, but it needs the outputs of the light reaction. The main products of the light reaction is ATP and NADPH. In dark reaction, the main products are glucose. It's a nature, photochemical. It's a kind of photochemical reaction. I told why it is photochemical. Here it's an enzymatic reaction. Why enzymatic? To fix the carbon dioxide to glucose, it need enzymes. That's why it's called enzymatic reaction. So coming to section 11.4. So how many types of pigments are involved in photosynthesis? That's very important question. The major pigment is chlorophyll A, but how many types? There are many types. Specifically, there are 4 types. So multiple pigments, not only one. These pigments absorbs different wavelength of light. हर एक pigment अलग अलग light को absorb करता है. And the leaf color is due to the combined effect of these all several pigments. So when you see the leaf, sometimes the leaf looks like a dark green, light green, only green, pale green. So it's all combination of different pigments which is involved in the photosynthesis. जितना भी पिगमेंट्स है उसका कॉम्बिनेशन का इफेक्ट वो लीफ का कलर दिखता है. So whatever the light the leaf doesn't absorb, it reflects. So usually the leaf doesn't absorb green light, it reflects the green light. That's why we see leaf as a green color. So if you take the paper chromatography, you know already paper chromatography you would have studied already in the chemistry is the separation of the pigments. There is solute phase and solvent phase. So if you put it as a solute as a pigments and if you separate them on the paper chromatography, you can see chlorophyll A as a bright or blue-green color is a very important chief pigment for the photosynthesis. And chlorophyll B, it looks like a yellow-green color. It is also an accessory pigment. Xanthophylls is also an yellow color. It's also an accessory pigment. Carotenoids, it looks like it appeared to be yellow to yellow orange. It's also an accessory pigment. So the important point here is the most abundant plant pigment in world is chlorophyll A. The most important pigment for the photosynthesis is chlorophyll A. So chlorophyll A is the primary pigment, very important pigment. Remaining all three are accessory pigment. It helps chlorophyll A. It transfers the light to the chlorophyll A. So chlorophyll A is a very important pigment. What are pigments? Pigments are substances that absorb light at specific wavelength. So any pigment which can absorb the light at specific wavelength is called pigment. So chlorophyll A absorbs light at separate wavelength. Chlorophyll B absorbs light at separate wavelength. Xanthophylls absorbs light at specific wavelength. Carotenoids absorbs light at specific wavelength. So each pigment has its own absorption spectrum. Spectrum is a wavelength of light. So कौन सा पिगमेंट कौन सा लाइट का वेवलेंथ को ऑब्जर्व करता है उसका ऑब्जर्प्शन स्पेक्ट्रम बोलते हैं. सपोज क्लोरोफिल ए है, ये ब्लू लाइट या रेड लाइट को ऑब्जर्व करता है. सो ब्लू एंड रेड लाइट इज ऑब्जर्प्शन स्पेक्ट्रम. सिंपल है. ऑब्जर्प्शन स्पेक्ट्रम इज द लाइट वेवलेंथ which is observed by the specific pigment is called as that pigment's absorption spectrum. Simple. I don't need to take like, oh, absorption spectrum ये बहुत बड़ा difficult word है. No, simple. Whichever the light the pigment absorbs is called as absorption spectrum. So how whichever light कैसा मालूम होता है? Wavelength के ऊपर मालूम होता है. So so absorption spectrum of chlorophyll A is very important. So this is, see, you can see the absorption spectrum of chlorophyll A that's in the green color. Chlorophyll chlorophyll A it's in the blue color, chlorophyll B it's in the green color, and carotenoids it's in the yellow color. This is the figure taken from the NCERT textbook. So the blue region, the peak absorption of the chlorophyll A is the blue region and red region. So you can see there is a one peak here. The blue chlorophyll A का एक peak इधर है, and another peak is here. So this is blue light. This is red light. So इसका नैनोमीटर कितना वेवलेंथ 430 नैनोमीटर 430 नैनोमीटर एंड रेड वेवलेंथ का रेड लाइट का 660 to 680 nanometer. Why it's coming between 400 to 700? Because we already know is between 400 to 700 is a visible spectrum. Visible spectrum के अंदर ये सब लाइट्स आता है. So 430 में blue light आता है, 660 to 680 में red light आता है. So blue light and the red light is the main absorption peaks in the absorption spectrum of chlorophyll A. देखो ये पूरा absorption spectrum है, इधर से इधर तक chlorophyll का ये absorption spectrum है. But इसमें peaks आता है, main जो which light it get absorbed maximum. That's called peaks in the absorption spectrum. The peaks are one is the blue light, one is red light. So again for chlorophyll B, similar to same. इसके नजदीक आता है एक peak that's also near to blue light and this is also near to red light. But for the carotenoids, more or less is near to the blue light. There is no another absorption peak for the carotenoids. Only there is a two peaks here in the blue light. So for chlorophyll A and chlorophyll B, there is a one peak each in blue light and red light, but in the carotenoids there are two peaks but only near the blue light. So the green region leaves appear greener because why I already told the green light is reflected. See, this is the green, the wavelength, agar if you take this one, this is chlorophyll A and this is the green ग्रीन कलर का वेवलेंथ. सो ग्रीन कलर का वेवलेंथ पूरा ये ऑब्जर्व नहीं कर रहा है. अगर ऑब्जर्व कर रहा तो इधर पीक आता था. ऑब्जर्व नहीं हो रहा है. दैट्स व्हाई इट्स रिफ्लेक्टिंग ऑल द ग्रीन कलर. सो ब्लू लाइट को ज्यादा ऑब्जर्व कर रहा है इसलिए इधर पीक आया है. रेड लाइट को ज्यादा ऑब्जर्व कर रहा है इसलिए इधर पीक आया. बट ग्रीन लाइट को रिफ्लेक्ट कर रहा है. जो लाइट रिफ्लेक्ट होता है वही कलर वो लीफ का आ जाता है. उसलिए ग्रीन लाइट रिफ्लेक्ट करता है ज्यादा, उसलिए लीफ ग्रीन दिखता है. एक हो गया ऑब्जर्वेशन स्पेक्ट्रम. ऑब्जर्वेशन स्पेक्ट्रम समझ में आ गया. जो क्लोरोफिल है कौन सा लाइट को ज्यादा ऑब्जर्व करता है उसको बोलते हैं ऑब्जर्वेशन स्पेक्ट्रम. दूसरा है एक्शन स्पेक्ट्रम ऑफ फोटोसिंथेसिस. एक्शन स्पेक्ट्रम मतलब ये होता है कौन सा वेवलेंथ में फोटोसिंथेसिस कौन सा रेट में रहता है. shows the rate of photosynthesis at different wavelengths. ब्लू ब्लू लाइट का अगर ब्लू लाइट का रेस आ रहा है तो फोटोसिंथेसिस कितना ज्यादा रहेगा, कितना बढ़ेगा. रेड लाइट में आ रहा है तो कैसा रहेगा, ज्यादा होएगा कम होएगा. ग्रीन लाइट आ रहा है तो ज्यादा रहेगा कम होएगा. उसको बोलते हैं एक्शन स्पेक्ट्रम ऑफ फोटोसिंथेसिस. सो इट शोज द रेट ऑफ फोटोसिंथेसिस एट डिफरेंट वेवलेंथ्स. डिफरेंट वेवलेंथ्स, suppose the 430 nanometer, 660 nanometer, 680 nanometer, 500 nanometer, कौन सा वेवलेंथ का लाइट में क्या का फोटोसिंथेसिस, कैसा फोटोसिंथेसिस रहेगा, ज्यादा रहेगा कम रहेगा मीडियम रहेगा, उसको बोलता है एक्शन स्पेक्ट्रम ऑफ फोटोसिंथेसिस. बट द कोइंसिडेंस इज ये ऑब्जर्वेशन स्पेक्ट्रम ऑफ क्लोरोफिल ए और एक्शन स्पेक्ट्रम ऑफ फोटोसिंथेसिस का दो भी मैच होता है. जो क्लोरोफिल ए ज्यादा रेड ब्लू लाइट को ऑब्जर्व करता है उधर ही ब्लू रीजन में मैक्सिमम फोटोसिंथेसिस होता है. दिस इज ऑल इंटरकनेक्टेड. सो अंडर द ब्लू रीजन, under the blue light the maximum photosynthesis happens. That's why the chlorophyll A observes more blue light. Under the red region the more photosynthesis happens. That's why chlorophyll A observes more red light. समझ में आ गया. So if you just take the peaks here, this is the action spectrum. This is the action spectrum of the photosynthesis. This is the chlorophyll A absorption spectrum. So you can see the two peaks here matching together. So there are some other peaks also here, but the two main peaks are matching together to chlorophyll A. इसका मतलब ये है ब्लू लाइट में ज्यादा फोटोसिंथेसिस होता है और रेड लाइट में ज्यादा फोटोसिंथेसिस होता है, उसलिए क्लोरोफिल ब्लू लाइट और रेड लाइट को ज्यादा ऑब्जर्व करता है. So these regions match absorption peaks of chlorophyll A. Chlorophyll A का absorption peak को मैच होता है. देखो chlorophyll A इधर है और ये absorption spectrum है. That's the rate of photosynthesis measured by O2 release. photosynthesis जब भी होता है O2 release होता है, उसके अकॉर्डिंग photosynthesis को मेजर किया जाता है कितना O2 रिलीज हो रहा है. तो इसको ओवरलैप किया तो देखते हो दोनों पिक्स इधर मैच हो रहा है. So chlorophyll A is a cheap pigment which is directly involved in photochemical reactions. All other pigments, as I told before, it transfers energy to chlorophyll A. जो कैरोटेनॉइड्स है, क्लोरोफिल बी है, xanthophylls है, वो लाइट एनर्जी को ट्रैप करके क्लोरोफिल ए को सेंड करते हैं. क्लोरोफिल ए में फोटोकेमिकल रिएक्शन होता है. सो क्लोरोफिल ए इज द चीफ फोटोसिंथेटिक सिगमेंट. ये जरूरत नहीं बस दोनों पीक्स इधर ओवरलैप हो रहा है, पूरा ऑब्जर्वेशन स्पेक्ट्रम और एक्शन स्पेक्ट्रम पूरा नहीं मैच हो रहा है, उसका पीक्स ओवरलैप हो रहा है. ऑब्जर्वेशन स्पेक्ट्रम का पीक और एक्शन स्पेक्ट्रम आपका फोटोसिंथेसिस का पीक, दे आर गेटिंग ओवरलैप्ड. Chlorophyll A का peak, only chlorophyll A. So the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis, the peaks are matching together. This is very important. This is very important for the NEET. So we already discussed about the chlorophyll A very well. So as I told, other than chlorophyll A, the chloroplast also has other pigments. That's chlorophyll B, xanthophylls, and carotenoids. These three are called as accessory pigments. So chlorophyll A is the chief pigment. These three are accessory pigments. Accessory pigments क्या करता है? It absorbs additional wavelengths. So chlorophyll A का blue और red light में ज्यादा light absorb करता है. Chlorophyll B ब्लू लाइट के थोड़ा बगल पे, रेड लाइट का थोड़ा बगल पे। xanthopils अलग वेवलेंथ में, कैरोटीनॉइड्स ब्लू लाइट और उसके बगल बगल में। सो डिफरेंट डिफरेंट वेवलेंथ्स का एनर्जी को, लाइट को ये ट्रैप करता है, क्लोरोफिल भी, xanthopils एंड कैरोटीनॉइड्स। दैट्स व्हाई इट हैज ऑल द पिगमेंट्स। सो अगर लाइट में क्लोरोफिल, ब्लू लाइट और रेड लाइट ही ज्यादा, हमेशा ऐसा ही नहीं रहता है कि द सनशाइन इज वेरी नाइस, the plant is getting only blue light and red light. There are sometimes the sunshine is not that much shiny or that much intense, so it has different wavelengths. The blue light थोड़ा कम हो जाएगा, रेड लाइट थोड़ा कम हो जाएगा, दूसरा लाइट अधिक मात्रा में रहेगा। तब ये पिगमेंट्स उसको फोटोसिंथेसिस होने के लिए हेल्प करता है। So it all takes the light energy from the sunlight and it transfers all the light energy to chlorophyll A. And further, this is very important. This all three pigments protect chlorophyll A from photooxidation. So what is the meaning of photooxidation? So when the chlorophyll A get reacted with oxygen, it forms free radicals. तो फोटो ऑक्सीडेशन क्या होता है? ऑक्सीजन जब किसी के साथ रिएक्ट हो जाता है और फ्री रेडिकल्स रिलीज होकर उसको डैमेज कर देता है, उसको बोलते हैं फोटो ऑक्सीडेशन। So when chlorophyll A get oxidized, it's reacted with oxygen. So chlorophyll A get damaged. So ये damage को chlorophyll B, xanthophylls, carotenoids prevent करता है. So this is very important. That's why these are called accessory pigments. It transfers its whatever the light energy is absorbed from the sunlight to the chlorophyll A, and it also protects chlorophyll A from photo oxidation. सो फोटो ऑक्सीडेशन में बोल दिया हूँ, और तुमको कैसा बोलूं? जस्ट लाइक क्लोरोफिल जब ऑक्सीजन के साथ रिएक्ट करता है, वो सिंगल ऑक्सीजन रेडिकल्स क्रिएट होता है। वो ऑक्सीजन, सिंगल ऑक्सीजन रेडिकल्स वो क्लोरोफिल को डैमेज कर देता है, तो उसको बोलते हैं फोटो ऑक्सीडेशन। So this kind of oxidation will also happen in when you burn oil, you burn fat, or this all, the it all create the free radicals. These free radicals are very dangerous to the health, ह्यूमन हेल्थ के लिए भी बहुत डेंजरस है। फ्री रेडिकल्स बहुत डेंजरस होता है, इसलिए जो भी ऑयल है वो काला हो गया है, बार बार उसको गर्म किया है, उसको उसमें उसके जो भी अंदर फ्राई किया उसको नहीं खाना है, क्योंकि वो फोटो ऑक्सीडेशन होके उसमें फ्री रेडिकल्स रिलीज होता है, वो हेल्थ के लिए हानिकारक है। तो उसलिए ये फोटो ऑक्सीडेशन होता है। फोटो ऑक्सीडेशन को प्रोटेक्ट करने के लिए क्लोरोफिल बेस्ड xanthophylls and carotenoids are very important. As I told before, accessory pigments expand the absorption spectrum. Chlorophyll का ये कौन सा absorption spectrum? Only blue light and red light. This is 430 nanometer. This is 660 to 680 nanometer, and it also prevent photooxidation. So neat final का क्या है? Chlorophyll is a cheap pigment. Blue light and red light is the most effective light for the absorption spectrum and also for the action spectrum. Accessory pigments protect and transfer energy to chlorophyll A.